you are very much the experts of this, and I am very much just starting this field, and I'm an immunologist, so I'd like to kind of share my view of pulmonary fibrosis. So IPF is tissue remodeling and extracellular matrix deposition, and the myofibroblasts plays a huge role in this. But where that comes from, is it a fibrocyte, is it EMT, is it proliferation by the resident fibroblasts? Those are still all active areas of research right now. We know that there's some kind of a lung injury to the epithelium event that happens, and this causes cellular mediators to be secreted, and the recruitment of a variety of different leukocytes, also secreting both pro-inflammatory and pro-fibrotic cytokines and chemokines. What I'd like to do is introduce a new cell to you called the innate lymphoid type 2 cell. And the way that it comes to the lung is in response to cytokines, and they're IL-25 and IL-33. And so this cell is kind of new on the docket, and where it comes from is very similar to where the leukocyte, or to the lymphoid cells come from. So we all know the paradigms of the Th1, Th2, Th17, Th240 million, but how it goes is they're characterized based on what they produce. Well, it turns out that in the innate immunity, there's also cells that have a very similar paradigm, and they are also characterized by their end products. So we have the ILC2 cells, natural killer cells. We have the ILC1, ILC2 cells, which are also called noocytes or natural helpers. And then there's this ILC3 group, which are kind of further characterized into the IL-17 and the IL-22s. So where they come from, we kind of have to follow the lymphocyte progenitor pathway once again, with the common lymphoid progenitor, the dominant or the double negative, the double positive with Notch signaling, then the CD4 cell, and the GATA3 is the major transcription factor that kind of pushes them towards that Th2 phenotype. Well, the ILC2 cell definitely comes, there's great work that shows that it comes from the common lymphoid progenitor cell, some work that suggests it comes from the double negative. But the transcription factors are ID2, ROR alpha, GATA3, and Notch signaling. And once again, they produce the prototypical Th2 cytokines. And so how do we find these cells? This is the big thing. There's no great marker, and basically the first thing we do is say what they don't have. And so they call them to be lineage negative cells, which means there's no T cell markers, no CD3, 4, 8, no B cell markers, B220, no erythroid markers, TERT110, no neutrophil marker, GR1, no CD11C, so no DC markers, no macrophage markers. They do have CD45 on them, yay. But the rest of these markers were ones that we really had never heard of or really not have thought about at all. And so these are large flow panels to be able to isolate these cells, and that's what we're starting to look at. Now, there's some evidence when I first put this proposal in that ILCs were important in pulmonary fibrosis. And part of this goes back to some of the original work that says there was good correlation between increased IL-13 production and IL-13 receptors with correlating with increased morbidity and mortality in IPF patients. And there was also some work that showed that alveolar macrophages, or I'm going to say a BAL cell, produced a lot of IL-13 in IPF lungs. There was also work that my PhD mentor did that showed that in an experimental fibrosis model, the FITC model, that IL-13 knockout mice were protected from developing fibrosis, but not IL-4, and that it showed that these mice had impaired collagen synthesis by the fibroblasts. Now, there was a couple other Th2 type diseases that had been previously showed to be important, the ILCs to be important. So in a model of allergic asthma, what they did is they took ILC2 cells from a wild type mouse and adoptively transferred them into an IL-13 knockout mice and experimentally induced asthma, and those mice were able to have similar asthma just like a wild type mouse. And so those cells, if those are the only IL-13 producer cells there, they can still exacerbate asthma. And there's also a lot of work with these noocytes, the ILC2 cells, in parasitic infections, another Th2. So based on that, we came up with this hypothesis that the ILC2 cells are major producers of IL-13. They can regulate, initiate the progression of pulmonary fibrosis. And as I alluded to, we are mouse researchers. I'm an immunologist. So we use an experimental fibrosis model of bleomycin with an intertracheal challenge and look at a 3-week time course where we're looking at ILC2 cells, specifically just by flow cytometry. There's actually a great antibody that we can deplete these ILC2 cells. We also were interested in measuring cytokines and harvesting lung macrophages. And so this was our really awesome preliminary piece of data where we isolated these cells, and these are lineage negative, CD45 positive, and we just used 2 markers to start out with, but CD90 and ST2. And we showed that these cells are present on day 0, but that after day 7 and day 14 after bleomycin, we see a significant increase in the amount of these cells after bleomycin treatment. Now, if we look at the amount of IL-13 that's being produced from these cells by intracellular flow, we see that it's significantly higher at day 7 than at day 0. So this showed that the IL-2 cells were there, they were getting there, and they were making the IL-13. Then this is the great piece of data. We know that if we give bleomycin and we look at day 14 and we measure the amount of hydroxyproline as a surrogate for the amount of collagen deposition in the lung, that it's significantly higher after day 14 of bleomycin treatment. But if I treat these mice with an anti-CD90, which depletes the ILC2 cells, there's a significant decrease in the amount of collagen in these mice. And so this was really what we were excited about and really kind of served as the preliminary data of this grant. And we were all set to write a great paper on this. And then there was some really exciting new evidence about ILC2 cells that came out in IPF. There was this paper that came out in December that said that IL-33 potentiates bleomycin. And basically, they gave IL-33 to wild-type mice after bleo, and they did much worse. They also showed that there was more IL-33 produced in ILD patients compared to controls. And then in January, this was a lovely paper from the Ireland group that came out that showed that in the lungs of wild-type mice, and this was using a schistosomiasis egg model of fibrosis, there were more ILC2 cells after the induction of an experimental fibrosis. However, when they did an IL-25 knockout mice or IL-17 receptor knockout mouse, there was no increase in the amount of ILC2 cells. So this really was what our data was. And so they actually did do one step further, and they looked in patients. And they showed that there was an increase in the amount of IL-25 in IPF patients. And they did something really interesting. They measured all the patients at one time, and then they measured them 1 year later. And they actually see that IL-25 significantly increases a year later. So this seems like this is really associated with more severe disease. And then they did flow cytometry, and they showed that there's increased ILC2 cells in the bronchoalveolar lavage fluid of IPF patients compared to normals. So it was a much better paper than what we would have put out, but we were getting there, and this really served as kind of the jumping off point that proved that this happens not only in experimental fibrosis, but it happens in IPF patients as well. And so I come back to this model. And we were looking at ILC2 cells, but this proposal was not just looking at the presence of them. I wanted to see what happens when they interact with other cells, because we know that the lung is not a sterile environment, and there's lots of other different cell types there. And what I've been interested in pretty much my whole career with lots of different avenues is the macrophage. And we know when the macrophage comes into an environment, when it sees the pro-inflammatory cytokines, like TNF, interferon gamma, LPS, it turns to this classical activation, or an M1. But if it comes into kind of a reparative phenotype with IL-13, IL-4, TGF-beta, IL-10, it actually turns into an alternatively activated macrophage, or an M2. And there's been a lot of work more recently that has shown that these macrophages are very profibrotic, and they produce a variety of different mediators, arginase, Fis1, YM1. And so in our model, we just wanted to verify that these molecules were upregulated. And so when we take lung macrophages from mice that were treated with bleomycin at day 7, we see a significant increase in all of these M2 markers. We also have some really exciting new data that has been with another project, but we were actually able to measure the amount of arginase expression in peripheral blood samples from IPF patients compared to normal healthy controls, and see a significant increase in the amount of arginase. And this has been under a little bit of debate, and so there's some people that say it's up, some people that say it's down, and we're excited to say that we really truly believe it's upregulated in IPF patients. So then I was also talking to Cheryl a little bit about my interest in macrophages. And I mean, when you think about it, there's an injury that happens in the lung. And so you're going to get recruitment of cells, and what those cells are coming into and what happens and how they get trafficked is not really understood or not really known. And so we have some preliminary data that says the resident alveolar macrophages in the open bars, they're pretty constant throughout the whole time course. However, the exudate macrophages, or the recruited cells, they start out, and at 2 days after bleomycin, we see a significant increase in the amount of those cells. It goes up about day 7, and then it falls down to baseline at about day 14. But what those cells are, whether they're M1, M2s, and really understanding how they're coming in and how they're differentiating and how they're being, what the phenotype is not known. And that's really what I want to spend the rest of my time with this grant looking at, is the role of ILC2s in mediating what's happening to these macrophages when they're coming into the lung. And so, there is one piece of data that has shown that ILC2 cells can regulate macrophage phenotype, and it's actually in a different model. But what they showed was that in gamma chain knockout mice, or in IL-13 deleter mice, which have reduced levels of ILC2 cells, when they measured the amount of arginase positive macrophages that were there, they showed they were significantly downregulated. So we really do think there is an interplay between macrophages and alternatively activated, ILC2 cells and alternatively activated macrophages. And so I leave you just with a little bit of a model. Once again, a very simplified immunologist version of what's going on. But we have some kind of an injury that occurs in the epithelial cells, and this causes recruitment of a variety of different cellular mediators. I've kind of highlighted IL-25 and IL-33 and the ILC2 cells. When they come in, they proliferate, and they make a lot of IL-13. So that way, when the macrophages come in after an injury, they come in and they turn into more of an alternatively activated macrophage, and they can also produce a lot of IL-13, which can definitely have detrimental results on the fibroblasts, with increased collagen production and proliferation of resident fibroblasts. And so I have to say this work is definitely a joint effort. And it started in the University of Michigan with my mentor, Ted Staniford, and I have a huge collaboration and still amazing relationships there. And now I am privileged to join the faculty of Ohio State with Dr. John Chrisman, and I have another great committee there. So I feel like I am kind of in the best of both worlds, with having these amazing scientists to be able to work with and collaborate with in multiple institutions. And I'm so excited to be able to add this line on all of my slides to say that I am now a InterMune junior faculty program, and thank you for your support. Oh yeah, anyone have any questions? Can I? Yes. Oh, I'm sorry.
So you know, very timely, as you know, there are anti-IL-13s that are in clinical trial right now. But what's intriguing to me, do you think the epithelial cells are sending the signal to haul in the ILC2s? And do these ILC2s feed back onto epithelial cell biology and repair? I think that might be a really interesting angle to study.
Well, I definitely wanted to look at the alveolar epithelial cells. Part of my other work has been really focusing on epithelial cells and their role in innate immunity, so I'm definitely interested in that. My understanding is right now the only marker, or the only cytokines that have been shown to really recruit these ILC2 cells are IL-25 and IL-33, and those are predominantly produced by epithelial cells. So I don't know exactly. And there could be, and I had actually put some co-culture studies in, but somebody decided to take them out, so. So yes, I'd like to go into that some more, for sure. Yes.
I remember looking at the work, and the thing that was most impressive was the unique nature of these cells and the conduction and the sequestration from the lung that made it specific and quite concerning the causative association. Comment on that?
I'm sorry, can you explain a little bit further? I guess I'm not sure. Like how it's just a lung-specific disease, or? Well, this actually, I mean, the work that I showed with the ILC2 cells, they actually first characterized them in a hepatic fibrosis model. And so I think this can be really put into a variety of different fibrosis models, not just lung-specific. Does that answer your question, or?
And that's my next point, that it appears to be fibrotic-centric.
Yes, definitely. Yes, definitely.
Do you know other epithelial cells that make IL-33? Is it general all epithelial cells?
I'm blanking on that right now. I don't know. I want to say probably that the liver epithelial cells will produce that as well, because that was the primary reason why we got interested in this. So I don't think it's a specific alveolar epithelial cell that only makes these. I'm sure that there's other cells that make them as well.
So you showed a very nice graph showing the somewhat transient nature of the exudate macrophages and the relative steady population of the resident macrophages. There's been data out there that recently that shows that the resident population can actually proliferate in and of itself. And so what do you think the role of the resident and the proliferation of the resident macrophages plays in all of this?
So I think what's hidden in that data is when you have injury, the first cells that are kind of come into contact with the bleomycin are the alveolar epithelial or the alveolar macrophages. So I have a feeling a lot of those ate up the bleo and died. And so the ones that didn't are the ones that proliferated. But somehow the cells are able to kind of maintain that still flat baseline. So I think there's a lot that's happening in the alveolar macrophages. And actually in another grant to a different place, I'm actually doing some chimera BMT experiments where I'm looking at a GFP marker, CD45.1, CD45.2, and looking at the repopulation of those cells and what's happening. I mean, when we do the turnover, are they for sure doing what I think they're doing, or is it something totally different? And so we're trying to answer that question in a different way by using some of these other markers. So we'll see how that goes.
Well, thank you. Very fantastic talk. And congratulations again. Fantastic. We'll go to the next.